We'll talk about we'll talk about two examples of one example of microbe-plant interaction. The other is a microbe-animal interaction. So for microbe-plant interaction, it's a characteristic of a parasitic interaction, whereas for the microbe-animal interaction, it is a symbiotic relationship. Okay. So for microbe-plant interaction, the example that we are looking at is the Tumorigenesis or crown gall disease by Agrobacterium tumefaciens. Okay, so this is just one example of microplant interaction. There are of course many examples, but this is well studied and it is of special interest to plant molecular biologists because of this TI plasmid, okay, tumor-inducing plasmid. So Agrobacterium tumefaciens is a gram-negative bacteria. Okay, it's special because it is its host range is wide. Okay, it can infect more than 600 species. So unlike certain parasites where it's very host specific, this one can infect a lot more plants, 600 over 600 species. Then the tumor-inducing plasmid. This is the plasmid that it has, and this is the plasmid that allows it to form the tumor. Tumorigenesis is a formation of tumor in plants. So in the TI plasmid, there's a tDNA portion. Okay, tDNA is the transfer DNA portion. So this is the portion that is transported and integrated into the plant genome. Okay. So now, after scientists have investigated this process, they have now applied their knowledge by using TI plasmid as a natural plant transformation system. Okay, so they are using the TI plasmid to transform plants. So basically, by inserting specific genes into the tDNA portion, you are able to integrate the gene into the plant genome. Okay, and this because the TI plasmid is from Agrobacterium tumefaciens, so it is a natural plant transformation system. Okay, so it has been used as a vector to genetically engineer plants. Example, the Bacillus thuringiensis toxin, Bt toxin gene of Bacillus thuringiensis has already been used to transform certain plants into having this Bt toxin. So Bt toxin is a natural a pesticide. Okay, so it's in the form of a crystal. So paraspora crystal, so it can kill pests. So if the plants have it, then it won't be susceptible to pests. Okay, so that's the importance of the TI plasmid in Agrobacterium tumefaciens. So this is TI plasmid. Okay. So this is the transfer DNA, tDNA portion. So this is the one that is integrated into the plant genome. So you please remember there are two specific genes here. One is a onc gene, this is the oncogene, and this is the OPS, opine synthesis gene. Okay. So opine synthesis gene will synthesize opines. Okay, so opines are carbon-based molecules, so they are actually uh, a form of food for the bacteria, because the Agrobacterium tumefaciens have this opine catabolic genes. So only the Agrobacterium tumefaciens can degrade the opines. So this is the portion that gets integrated into the plant. So the tDNA tricks the plant into producing opines, which can only be used by the bacteria. Okay, so this is an example of a bacteria tricking the host system into producing food for the bacteria. Okay, so the oncogene is oncogene. So when you talk about oncogenes, you of course relate it to cancer, and cancer is basically the unregulated uh, replication of cells. So it's the same situation here, where this gene will promote cell replication, the plant cell replication. So not only does the tDNA tricks the plant into making more opines, it tricks the plant into making a lot of cells that will produce opines. So the amount of opines that is synthesized increases. Okay, so this is how the tDNA is integrated into the plant genome and tricks the plant into making more cells, more opines for the bacteria. So these are the virulence genes, genes. So these are the virulence genes that will help the integration of tDNA from the bacteria plasmid into the plant genome. Okay, so let's look at the. So for an infection process to occur, the plant has to be injured. The injury can be a cut by animals or by humans or even a natural process. So injured plants will secrete, because if the plant is not injured, it'll have a thick layer of bark. Okay, and then with that thick layer of bark, the bacteria do not have a chance to attach and begin its infection process. So similar to humans, our skin is our most important defense mechanism. So long as our skin is intact, we are not so susceptible to diseases. Okay. Uh, so similarly, that's why if you have a cut, a deep cut, once that you can expose your flesh, then you shouldn't go swimming, and you shouldn't expose it to uh, the environment, because the chances of infection is there. Okay, so the injured plant secretes. Plants that is injured will secrete a phenolic inducer. Okay? So this is a not normal process that is carried out by plants. So it secretes a phenolic inducer and stimulates bacterial attachment. Okay? So the bacteria will attach to the plant, and then will activate the. And when the bacteria attaches, this will activate the Virulence genes. So when the Vir gene is activated, the Vir will activate the Vir by phosphorylation. So Vir is activated because Vir is located on the bacterial cell membrane. It is a transmembrane gene, protein, so it is activated first. Okay, so once it's activated, it will next continue activating the Vir by phosphorylation. Then the Vir will initiate the transcription of other Vir genes. Can you hear me?
Yes, doctor.
Yes, it's fine.
Can you hear the rain?
No.
Okay, that's good. Okay, so among the Vir genes that is initiated by Vir, first is the Vir. So Vir will create a single-stranded nick adjacent to the tDNA. Okay, somewhere here. So that nick, once you have a single-stranded nick, you can start the rolling pin mechanism where the tDNA, the single-stranded tDNA is released from the TI plasmid. So please, if you're not familiar with the rolling pin mechanism, to look back at your, probably your recombinant DNA mechanism will have, your recombinant DNA technology course will have this. Okay. So once the tDNA is released, it's single-stranded, so it's not stable. So you have to have the Vir that acts as a single-stranded binding protein to stabilize the tDNA, to stabilize the single-stranded tDNA. Okay. So once the Vir is attached to the single-stranded tDNA, then the Vir will form the conjugation bridge. So this conjugation bridge will allow the single-stranded tDNA migrating from the bacterial cell into the plant cell. Then once it enters the plant cell mechanism will integrate the single-stranded tDNA into the plant genome. Okay. So this is how the infection process occurs. So Agrobacterium tumefaciens is a classic example of a parasite. Okay, because the integrated tDNA has the ONC and the OPS genes. So the oncogene here produce auxin and cytokinin. So these two are the plant growth hormones. So these two will induce unregulated growth of the plant cells. Okay, so this is also called tumorigenesis. Then the OPS allows for the production of opines. So the opines is a molecule that has both carbon and nitrogen in it. So a good source of food for the bacteria. So this is a classic form of parasitism where the Agrobacterium tumefaciens hijack the plant mechanism to produce food for the bacteria. Any questions?